Okay, in this video, demonstration video on F1 flow cytometry analysis, we're going to look at some basics of data clustering and other aspects related to that, and looking at our data further, identifying clusters. So what we're going to look at is common methods for exploratory data analysis with R, and think about how we use different dimensionality reduction methods, that are typically correlation based, clustering methods, and looking at markers using heat maps and other plotting functions. So as you know, there's certain packages like Flow Sum and Phenograph that do unsupervised clustering to split the cells into groups. And one consideration is that with TSNE and UMAP, garbage in, garbage out, and that if you have data of a particular shape, they may not necessarily resolve it in an appropriate manner. Other methods like PCA or MDS may be more reliable or more easily interpretable by people, whereas UMAP and TSNE sometimes lead to misinterpretation. So what we're going to do is install this package and create a matrix so you can apply TSNE to that. So let's switch over to R to try this. So what we need to do is install the TSNE package. You don't need to restart your computer. You may find there's a number of dependencies that install there. Then you need to activate the TSNE package, not tidyverse. And then what we're going to do is create a data set called data TSNE using the rnorm function like we did before, the mean of 1 and a standard deviation of 2. So if we just do a histogram of data TSNE, we can see that it has this shape. So it's a very uniform distribution, just has some noise. We can create a matrix of this data, and remember with matrices, you can also plot them in the same way as before. So we get the same distribution if we plot all that data. So we're making that data with 100 rows and 100 columns because we have 10,000 data points. So it's kind of a matrix with some noise. And if you look further and do histogram of any particular subset of that, you'll find that relatively speaking, get a fairly normal distribution. It just might have more noise if you subsampled it more. So any differences are just subsampling issues. What we can do is we use the RTNSE normalized output to normalize this matrix. So we're calling that norm TSNE, and we can plot a histogram of that. And in the same way as we had before, we can plot subsets of that to see that they have pretty much similar shapes. Just that might sometimes differ a bit. But you see that the scales, they're mean centered and rescaled. That's what this normalization function does. So the mean is now 0, whereas before for the matrix it was just centered around 1 or so, based on our initial function. So now we can use the RTNSE function with a perplexity of 1 and 2 dimensions to create a 2-dimensional plot. So we're sending this data from this function, from that input data to this function, to this plot TNSE variable. So plot TNSE is a list containing this data, and that's why we can use $y to extract the information from that list that contains the points for the TNSE plot. So you can see there we have those points listed here. Are pulling one vector against the other vector. So if we run this, what we see is that we get this kind of plot. So what that perplexity factor of 1, that seems to show different clusters. Let's try a different perplexity factor. So we've got a perplexity of 10, 2 dimensions, and we're going to plot it in blue. And what we see is more of a uniform distribution. In fact, if we rerun this code with a perplexity of 10, we can see each time we run it, we actually get a different result, which is interesting. So it's a different cloud set. And if we look at the these values, again, we get differing outputs each time we run it. And if we try with a perplexity of 20 again, and we're coloring this brown, you can see we get more of a mixed spread, but different colors each time. So what we can do is change these colors even. Let's see if this works. Let's try 3 colors. So you can see there's different colors there. We can see how those colors are spread throughout here. So you can see we get a mix. So what you can do is make your own data set and use TNC to plot the differences between different groups. And what's noting here is that as you change your complexity, you get different clustering levels. Oh, that's too high. See for a complexity of 25, you can see that the clusters are lost. If we change that to a perplexity of 5, again, we get partial clusters. So if we change it down to 2, you can see you tend to get different groups. We know this is just noisy data. It does not have any real underlying features. So the complexity has to be a positive number. You can see there, it's worth noting how the parameters that we put into TNSE can affect the extent to which we have different clusters. And what's important here is that just because we have different groups does not mean that they really are biologically valid. And secondly, because we're mapping any dimensions onto this space, the distances between these different groups may not be meaningful. And we can see that because if we rerun this code, we get a different arrangement each time. Okay, so that concludes this part. We'll discuss this further in the lab.